Good afternoon, everyone. So, as Marianne mentioned, this topic is looking at rosy gold apples. And here we looked at the different maturation periods as well as canopy positions and how this relates to your post-storage quality. Just a heads up, this is an ongoing project, but I will share some preliminary data in the meantime. So just some rationale behind the project. We all know that starch breakdown is an important parameter to determine your optimum picking period. And you're often aware with the starch iodine test and the staining, and then if it stains dark, that means that starch is present. However, the starch charts are subjective. You need a trained eye in order to interpret it, especially differences between cultivars. So certain cultivars stain different pattern, more radial staining, so it becomes difficult to match up to your specific starch breakdown. Another issue is, or challenge is that it often stains gray. So one isn't sure that should you actually count it as starch or not. So the staining of the gray does make it a bit different or a bit challenging. So what we thought about, maybe we should then quantify how much starch is actually present. And then we looked at your starch content in relation to your starch breakdown percentage. So some of the objectives we looked at was your relationship between your starch breakdown and your starch content, the rate of starch breakdown and starch content, as well as the variability within the tree canopy. We then related this starch content to your post-storage quality. Look at the materials and methods. We used rosy glow apples in the algae production region. We then sampled at 4 different canopy positions, inside out, bottom inside out, as well as we sampled weekly before commercial harvest. And here we collected all our samples, the maturity parameters, and then currently our starch content analysis is underway, so I'll just show some maturity data in the meantime. Looking quickly at our starch breakdown percentages, and this is at the different canopy positions, we saw that our outside top fruit had a higher starch breakdown, while generally our inside top fruit actually had a lower starch breakdown. There's similar trends for both seasons. When we combined all the canopy positions together, we saw that in season 1, we actually had a faster rate of starch breakdown, where in season 2, we had a slightly lower rate of starch breakdown. So the question is, I wonder if the starch content was actually the same between the 2 seasons. And interesting enough, we look at the sugar levels, our season 1, which had the fastest starch breakdown, actually had a lower sugar levels compared to the second season. So again, this relates to your starch converts to your sugars, so actually what was the initial content. Similarly to your malic acid, we saw that season 1 was lower, so initially started off with the lower acid levels, and then that also decreased faster in your season 1. Just to summarize the maturity, and we looked at the different canopy positions, we saw that your outside top fruit was more mature, so more yellow, larger fruit, better color. Our inside top fruit, however, was softer, but had a lower starch breakdown. So there's a bit tricky because it's contradicting. One shows it's more mature and the other says it's not. So that could be the subjectivity of your starch breakdown percentage. It could have been read wrong, or it was your starch content that could have different. So hopefully we'll have that answer, so get a better picture later on. When we look at our inside bottom fruit, we found that to be less mature as well. So just to say thank you for WorldGrow for funding the project, as well as Experico colleagues, and then Fruitways for the trial site.